Right. Thank you very much. People can just take their seats, please. We'll have people streaming in, probably continuing. We have several hundred people actually across the hall also watching on the video screen. I wanted to begin with a land acknowledgement. At SickKids, we would like to begin by acknowledging the land on which SickKids operates. For thousands of years, it has been the traditional land of the Huron, Wendat, and Petun First Nations, the Seneca, and the Mississauga of the Credit River. Today, Toronto is home to Indigenous peoples from across Turtle Island. SickKids is committed to working toward new relationships that include First Nations, Inuit, Métis peoples, and is grateful for the opportunity to share this land in caring for children and their families. We're having a very special symposium here today entitled The Future of Homo Sapiens, Technology, Science, and Health. It's a rather grand title, but it drew in quite a few of you. So thank you for joining us today. We have a scientific meeting here over the next 2.5 days that's being sponsored by Nature and Nature Medicine, and you'll hear from our editor from Nature a little bit later. But by way of introduction, my name is Steve Scherer. I'm the Chief of Research at the Hospital for Sick Children and also Director of the McLaughlin Center and a university professor at the University of Toronto. This event tonight is being sponsored by the University of Toronto's McLaughlin Center and also the SickKids Research Institute. This is the agenda for tomorrow. Some of you who are speaking tomorrow morning for the Nature Medicine meeting will see your names here. And for those of you who are not scientists, you probably don't know, but quite often when we have international meetings, we have public forums to discuss critical issues of the time to make sure we're getting public input and sharing information with those people who support us. So that was the initial rationale for putting this meeting together this afternoon. And around the same time, when we started to organize this meeting about 12 months ago, I was reading this book by Yuval Noah Harari, which many of you I'm sure have read, called Sapiens, Adapted from the Brief History of Humankind. This is from the very cover story in this book. And I was struck by the topics that we were covering in the meeting over the next 2.5 days actually came up in his list of important events in human history. So I thought this is slightly adapted from the book, but it's actually very, very similar. So I thought I would take a moment just to go through these adapted events to come to set the stage for the rationale behind this meeting. So 2.5 million years ago, we evolved as the genus Homo in Africa, and stone tools were first used. 300,000 years ago, we started as a species to use fire. 70,000 years ago, the emergence of language, which I would argue is the most important event in human history. 13,000 years ago, Homo sapiens, and Homo sapiens in Latin is human, with the sapiens being defined as wise or astute, remains as the only surviving species on the planet. So Neanderthal and Floresiensis and the other hominin species had all disappeared 13,000 years ago. 12,000 years ago, agriculture evolved. 500 years ago, the scientific revolution. 200 years ago, industrial revolution. And roughly 100 years ago, nuclear physics, medicine, and also we leave the confines of our planet, so space travel. And then what the author goes on and talks about is he uses the term intelligent design in his table, but if you go back and read the book, he defines this really as advances in what we're discussing here in the next 2.5 days, which is genomics applications to health, engineering and health, artificial intelligence, and precision medicine. So I thought, what a perfect venue to try to bring the world leaders to Toronto to host this public symposium. So I think this is going to be a momentous day. We really have the three leaders in the world in these three areas. So the rationale for the meeting is to be astute. The founders of the disciplines of genomics, AI, and precision medicine may all be in Toronto. So I should try to invite them to tell us of their vision of the future. What future generations will learn in school 50 years later. Experiments to do and experiments perhaps not to do. I have not told the speakers what to talk about, which could be dangerous, but we'll see. I did say that you might want to talk about things that you would like to see done and perhaps things that you don't want to see done. That's the only ground rules here tonight. And the second is to be wise. I wanted to remind everyone that scientific advance comes by exploring our curiosity and testing ideas through research. And everything you hear tonight started in some form with fundamental research. We're in the Research Institute at the Hospital for Sick Children. This is one of the great teaching hospitals of the University of Toronto. We have other incredible institutions across the city, making Toronto really one of the top world sites for medical research and fundamental research in the world. And we're here to celebrate that tonight. So who are the people I wanted to invite? This is really a list of my favorite scientists in circa 2024. Our first speaker will be Homo sapiens, Geoffrey Everest Hinton. Jeff, as a joke. Jeff is a distinguished university professor at the University of Toronto. And I'm just going to read his citation for his 2018 Turing Award for conceptual and engineering breakthroughs that have made deep neural networks a critical component of computing, including contributions to backpropagation theory, Boltzmann machines, neural networks, and also very importantly, including many gracious acknowledgments he makes to his many trainees and collaborators that have really revolutionized our understanding of AI. I am a geneticist, so I couldn't avoid showing his family's pedigree here on the left. It's on the internet, so it's not private information. It's a nice picture of Jeff, if you can see it there as a child. And his family tree is quite impressive. I would say I haven't seen one quite like this, going back to George Boole from Boolean logic. I think it's the foundational ideas of computer science, the Everest family from Mount Everest. He does have, I think it was an uncle who invented the Jolly Jumper also. So it's really quite incredible pedigree here. And there's been books written about Jeff's work recently on the bottom right. And I did want to just, for each of our speakers, point out my favorite scientific paper. This one of Jeff's came in Nature, where he discusses learning representation by backpropagation errors. And I don't know if Jeff remembers, but about a decade ago or so, we hosted a CFAR event, and he explained to the audience that he wanted to understand how neurons and cognition worked. And to do that, you really need to establish the field of artificial neural networks, which is comprising artificial intelligence. So he defined the biological problem and invented a system to actually dissect that problem. I think this is the beauty of science. So thank you so much, Jeff, for participating. Our next speaker is Homo sapiens, J. Craig Venter, known to all of us, certainly in the genetics community. I'll read his President's Medal of Science citation from 2008. For his dedication to the advancement of science of genomics, his contributions to our understanding of its implications for society, and his commitment to the clear communication of information to the scientific community, the public, and policymakers. These are photographs of him with President Clinton at the announcement of the first draft of the genome sequence back in 2000, the turn of the millennium, and also winning his award with Barack Obama. And quite interesting, if you haven't read this book, I highly recommend it. It's the only book I know of a human being. Craig, of course, as he'll tell us, was the first human identified to have his human genome sequenced, which he did himself. And but he kind of describes in this book not only his biological history, his genetic history, but all of his experiences and exposures in life that made him the unique individual that he is. We can debate this, Craig, but my favorite paper of yours was the first random genome sequence of Haemophilus influenzae. This is the first genome sequence of a living organism. And what is unique about Craig, while all the rest of us in the community were looking at one gene at a time, taking a bottom-up approach. If you think of a jigsaw puzzle, he was taking one piece out of the box at a time and trying to piece them together. That's what we were doing. Craig came up with the idea of looking at holistically everything at once. So shotgun sequencing all the DNA fragments and then using high throughput computational to piece together the jigsaw puzzle of life. Of course, he did this in 1995 for Haemophilus. He went on to do the Drosophila genome, which is about 1/20th the size of the human genome, the mouse genome, which is equivalent size to the human genome, human genome as part of the genome effort at Celera Genomics, and then his own genome sequence. He sequenced genomes across the oceans, and I think is the first individual to synthesize life de novo. And I'm kind of hoping he talks about that, but I didn't tell him what to talk about. Thank you for joining us, Craig. And last but not least, Homo sapiens, Kathryn North. This is a beautiful picture of Kathryn at the Murdoch Children's Research Institute with some of the patients that come to the hospital there in Melbourne in Australia. And I'll just read her citation from the Companion of the Order of Australia. For eminent services to genomic medicine, the topic of the meeting the next 2.5 days, nationally and internationally, to medical research in the fields of genetics, neurology, and child health, and as a mentor and a role model. So you'll see this through all three of these participants tonight, that mentoring, building teams, and passing on the torch is a key part of their careers. Kathryn has lots of great papers. This is the one I think is her highest cited paper, and also one that I read earlier on in my career. And I think she may touch on this tonight, but let's see. She used genetic approaches to, based on genotype or genetic approaches, to find associations with elite athletic performance. So she studied the DNA of sprinters and could show that specific genetic variants could be associated with speed. Now, that's what Kathryn has published, but I do think that her legacy will truly be her role in setting up the Global Alliance for Genomics and Health Research. And this initiative is a worldwide effort spanning all of the major countries to set up infrastructure to and ideas to share genomic information to benefit humankind as a whole. And I think she'll probably touch on that. We did have the International Precision Child Health Initiative meeting here in the last day and a half. And we also have representation from Boston Children's Hospital, the Great Ormond Street Hospital in London, and of course, Kathryn's Hospital in Melbourne, and SickKids. And then, so we're going to hear from each of these participants consecutively for 30 minutes each, up to 30 minutes each. And we have two distinguished panelists joining us, and then we're going to come to the front and have a discussion. The first is Janice Stein, Professor Janice Stein, and she's also, like Jeff and myself, a distinguished University of Toronto professor. And when I was thinking about who to want on the front of the panel, I said, we need someone who's just as smarter than the speakers, and Janice is the answer. So Janice, thank you so much for joining us. She's also the director of Munk School of Global Affairs and Public Policy. She's not a scientist, but she's going to put everything in perspective for us. And then secondly, Darren Burgess, who's a PhD senior editor from Nature magazine. For those of you who don't know what Nature is, it is the oldest scientific periodical in the world. And it is considered one of the top, if not the top periodical to strive to publish your work in. And they're co-sponsoring the meeting. And we're so happy to have you here, Darren. I was looking at this, you grab all these pictures off the internet, and I was looking at this photograph of Darren, and something just, I couldn't figure out, but something caught my attention here, and I kept going, did some work, came back to the photograph. I said, what is it about this? And then sure enough, you had stuff on your desk, which is dangerous. And I looked at the cover of the Nature issue he was working on, and it's actually our scientific paper. So I went upstairs and I looked, I said, that's actually our paper. And so I thought that would bring us full circle back to my opening comment of who I was. And just to leave you, you have to be careful what you leave on your desk. So with that, I'm going to turn the mic over to Geoff Hinton and invite him to share his vision of what he thinks is important in science for the next 50 years or whatever. Geoff, you can talk about whatever you want.
So the main thing I'm going to talk about today, the main thing I'm going to address is the issue of whether when these large chatbots say things, they actually understand what they're saying. Because that's very important for the future. Are they like us? Do they actually understand what they're saying? Just 3 slides of history. A long time ago, there were 2 paradigms for intelligence. There was the logic-inspired approach, which dominated AI for 50 years and didn't work. And there was the biologically inspired approach. In the logically inspired approach, the idea was you've got to figure out how things are represented in the brain or in a computer. and learning will come later. In the biologically inspired approach, the idea was you have to figure out how the connections in a neural network are learned, how you learn the synapse strengths, and things like reasoning come much later. So a neural network, the artificial ones, you have layers of neurons in the simplest ones. You might put an input image in the bottom, and you might want to get a decision at the top about whether it's a cat or a dog. And there's a very simple way to train it that's obvious to anybody who knows anything about evolution, a sort of very simplistic view of evolution. What you do is you take one of the synapses, one of the connection strengths, and you change it a little bit, and then you see if the thing works better. If it works better, you keep that change, and if it doesn't, you wouldn't, or you make the opposite change. Now that works, and you don't actually have to know how the network works for that method to work. It's just incredibly slow, because you have to do experiments where you change one synapse at a time. There's a different approach, which relies on the fact that you know how the neural network works. And what you do is you put the image in the bottom, you get your decisions at the top. So to begin with random, it might say 50% is a cat and 50% is a dog. And you'd rather it said, if it's a dog, you'd rather it said 49% is a cat and 51% is a dog. And if you compare the answers you get with the answers you like, you can send a signal backwards. And using calculus, you can figure out for every connection in the network, all at the same time, whether you want to increase its strength a bit or decrease its strength a bit to get a better answer. And that's more efficient than the random mutation method by a factor of the number of weights in the network. So if the number of weights in the network is 1 trillion, it's a lot more efficient. And that's how all the neural nets are trained. In 2012, a group of 2 of my students, basically, at the University of Toronto, showed that these deep neural nets are just much better than standard computer vision for recognizing objects in images. And that opened the neural network floodgates. Before that, many of us knew that neural nets were going to work better. But it was at that point that everybody switched to doing neural networks within a few years. And then neural networks became AI. And all the AI people said, oh, we want funding, it's AI. But for many years, neural networks had not been AI, they'd been the opposition to AI. People also said, but what about language? These multiple layers of features are good for recognizing objects, but they won't work for language. There's some quotes on my webpage. The linguists were extremely skeptical. There's a school of linguistics that comes from Chomsky, which is completely crazy and thinks that language isn't learned. If you want to cult, what you have to do is persuade people to say something that's obviously false, like my crowd was bigger than Obama's crowd. And for linguistics, the equivalent is to say language isn't learned. We were just the opposite, the neural network people. We hoped that a big neural network that started with random weights would learn both the syntax and semantics of language just by trying to predict the next word. And it turned out we were right. So here's 2 very different theories of the meaning of a word that have been around for a long time. The structuralist theory, which is what the symbolic AI people believed in, is that the meaning of a word comes from its relationships to other words. A word occurs in lots of propositions, and its meaning depends on how it occurs in those propositions. And that's sort of correct. But there's another theory that's also sort of correct, which comes from psychology, which is the meaning of a word is just a great big set of semantic features. And words with similar meanings have similar semantic features. And those 2 theories look to be completely different, but actually you can unify them. So in 1985, I made a little language model, actually a tiny language model, which unifies those 2 theories in the following way. You convert words into vectors of features in a neural net, and you use those features to predict the features of the next word from the features of the previous words, and you train the whole thing by backpropagating the error you make in predicting the next word. And hey presto, it will learn semantic features for words, and it will learn how those features should interact with one another to predict the features of the next word. And so what's happening now is the knowledge that the neural network has is not in stored sentences or propositions. It doesn't store any sentences or propositions. The knowledge is in how to convert a symbol into features and how those features should interact to predict the features of the next word. And that's how all these big language models now work. And notice it's very different from the symbolic AI view, which is knowledge will consist of strings of symbols. Now, we applied later on this kind of model to AP Newswire stories from the 1990s and looked to see what features they gave to words. Now, you'd see a big pattern of neural activity which wouldn't mean anything, but we could ask the question, show me nearby words that have similar features, and show me a bit further away words that have less similar features. and we invented a way of displaying words in a map. So the closest ones will be the most similar ones. And here's a little bit of the map. I hope you can read this. At the top, there are US states. Then there's some cities like Philadelphia and Detroit and Toronto. You'll see Toronto is quite like Detroit, but also quite like Boston and Melbourne and Sydney. You'll see on the right there, there's a city that is actually Cambridge, exactly on top of Oxford. As far as the net's concerned, they're identical. You'll also see, this goes down better in Montreal, but you'll also see that Quebec is actually not in North America. Quebec is next to Berlin and Paris. That should make the separatists happy. If you look at the bottom, you'll see that Israel, Vietnam, and Iraq are more or less identical. The large language models we had today, I think of as descendants of this tiny language model. They work the same way. They use many more words. They use many more layers of neurons. They use much more complicated interactions between features. And if you look inside them, with the little language models, you could see how they were working, and nobody doubted that they were getting the right features and understanding what was going on. But the big language models, you can't really understand how they're working by looking inside them. You look at the weights and you look at the features, and it's all too complicated. So it's very hard to know if they really understand what they're saying, and if they really are intelligent, or whether they're just somehow getting statistics from language and regurgitating stuff. Well, the answer is they really are intelligent. And you can see that by giving them a reasoning task that they've never had before. So there's a very good symbolic AI guy at Toronto who's retired now, who really believed in symbolic AI and was one of the best people doing it. And he suggested a problem to me. I made the problem a bit more complicated. And so you tell the neural net, the language model, the rooms in my house are painted blue or white or yellow. Yellow paint fades to white within a year. In 2 years' time, I want them all to be white. What should I do? And when I gave it that problem had never been on the web. And I gave it to the version of GPT-4 that couldn't look up things on the web. It had just had all its knowledge within its weights. It didn't have any stored propositions. It couldn't look on the web. Just the weights of the neural network were all the knowledge it had. And what it said was, assuming the blue paint does not fade to white. Now, when you give this problem to people, most people don't actually put that in. They do assume that, but they don't actually mention it. And you want all the rooms to be white in 2 years' time, here's what you do. And it gives exactly the right answer. The rooms painted in white, you don't have to do anything. The rooms painted in yellow, you don't need to repaint those because it's going to fade to white within a year. And the rooms painted in blue, you need to repaint with white paint because blue paint will not fade to white on its own. It understands things like if you fade to white, you don't then later fade to something else. It understands lots and lots of things like that. Okay, that's my evidence that it really does understand. Some people say, the symbolic AI people say, they don't really understand because look, they have hallucinations. They say things that are false. But so do people. The fact that they do that just makes them much more like people. Just like the LLMs, we store knowledge in synapse strengths. And when we remember stuff, we're not getting a file out of memory. We're synthesizing stuff. If it's about things that happened recently, the weights have lots of information about what happened recently, and we synthesize the right answer, pretty much. If it's about old things that we haven't rehearsed, we synthesize stuff that seems plausible to us based on lots of stuff we learned since the events happened. And we typically get all the details wrong. Judges don't like juries to know that. We're often very confident about the details we get wrong. And there's a wonderful example of it, which is John Dean's memory. Because you normally don't know what the ground truth was. But John Dean testified under oath at the Watergate trial about conversations in the Oval Office without knowing they were recorded. And most of what he said was wrong in detail. He said this person said that, it was a different person said that. That person wasn't even in the meeting. He said there was a meeting of these people. Those people never were in a meeting. But he wasn't lying. He was saying what seemed plausible to him. That's what a memory is. It's what seems plausible to you. There's no difference between a story and a memory. A memory is just a story that seems very plausible given what you've learned. And he was actually getting the truth. That is, what he was saying conveyed exactly the gist of the scheming that was going on in the White House. Now, chatbots are currently worse than people at confabulating, but getting better all the time. But what I want to emphasize is the fact that they confabulate shows they're more like us, not less like us. So the conclusion so far is that these large language models are very like people. They represent knowledge as interactions between learned features. just like we do. They generate memories rather than retrieving stored copies, just like we do. But now I want to come to a way in which they're different, and it's to do with how you share knowledge. So I'll talk about 3 different ways of sharing knowledge. There's the method that the symbolic AI researchers believed in. This is also the method that really bad teachers believe in. And the way it works is this. I've got facts in my head, and I take the facts out of my head, put them in sentences, and plonk the sentences into your head. And maybe if I do it many, many times, it'll eventually get into your head. But the idea is that knowledge is in these propositions, these symbolic propositions, and I have to get them from my head to your head. There's what's actually happening now with these neural networks. The way these big neural networks get knowledge from us, and the way we get knowledge from each other, and the way 2 big chatbots will get knowledge from each other if they had different internal architectures. It's called distillation. And it works like this. I listen to you saying something, and if I trust you, I try and change the weights in my brain so that I would tend to say the same thing. So what's happening is I'm not storing the sentences. I don't store the sentences you say. I change weights so I'd be likely to produce similar sentences. Similarly with your behavior. I look at your behavior, and if I want to copy you, I change the weights in my brain so that I will produce similar behavior. Now that's great, except it's very low bandwidth. If you think about language, you only get a few bits per word, 10 bits per word if you're lucky. Actually, we know the numbers. It's about 6 bits per word on average. If you're a politician, interestingly, it's about 5 bits per word, or 4 bits per word. A researcher I know who worked on this earlier said politicians are 4 bits per word because they're trying very hard not to say anything. But anyway, it's a very low bandwidth channel. There's one other way of sharing which we don't have. If you happen to have many different copies of exactly the same neural net running on different pieces of hardware, they can all go off and look at different bits of the internet, and they can figure out how they would like to change their weights to absorb the information that they've looked at, and then they can average all those weight changes. And now every one of them knows what all of them learned. That's a much more efficient way of training. Imagine if you could take 10,000 people, they could do 10,000 different courses, and as they're doing these courses, they're exchanging information with each other, and at the end, they all know what's in all 10,000 courses. That would be great. But we can't do that because we're not digital. You have to have models that use their weights in exactly the same way to make weight averaging make sense. So digital has a huge advantage. They can share knowledge by averaging weights. And that's got a bandwidth. If they've got 1 trillion weights, that's got a bandwidth of trillions of bits each time they share weights. It's just hugely more bandwidth. And that's why GPT-4 can know so much more than us. You can have many copies of it running on different hardware. They can all look at different bits of the internet and learn different things. And then they can all convey that to each other by sharing weights, by sharing actually the changes they're about to make to the weights. And that makes digital intelligence far superior to analog intelligence, which is basically what we've got. That's why GPT-4 knows more than thousands of times more than any one person. It's a not very good expert at more or less everything. So what do I conclude from that? Digital computation requires a lot of energy. People are talking about building nuclear power stations per data center. But it allows digital agents that have exactly the same model of the world to share extremely efficiently. We use much less energy because we're analog, and that's a more efficient way to do the computations, but we can't share efficiently. We have to share in this very clumsy way using language. So digital computation is just better. And digital agents are going to be much smarter than us. And that leads to a question, which is a big question for the future, and nobody knows the answer to it. Can we find a way to ensure that digital agents that are much smarter than us never want to take control? And the analogy I have for this is we're like someone raising a very cute tiger cub. And raising a tiger cub is just great, but you better be damn sure it never wants to harm you. And on the whole, raising tiger cubs is not advised. Now, I've managed to talk quite fast, so I do have a few more minutes. I've actually got about 10 more minutes, so I can say something else. So what I'm going to say now is going to make you think I'm extremely flaky and not believe anything I said so far. So there's a sentience defense. So people tend to think they're special. They think they were made by God and he put them at the center of the universe. Many people think we've got something very special that a machine could not have. So many people are not materialists. And this special thing is sentience. There's many versions. There's consciousness, there's sentience. I will focus on subjective experience. They think that a computer couldn't have subjective experience. That's how we're different from computers. We have subjective experience, and these machines don't. So I'm going to put forward a view called atheaterism. Dan Dennett sanctioned the name. It's basically Dan Dennett's view. But most people's view of the mind involves an inner theater, and only you can see what's in the inner theater. And that view is as wrong as a religious fundamentalist view of the material world. And nearly all of you hold that view. You think the mind is some kind of inner theater in which there's things that you see, and that's what subjective experiences are. And that's a mistake. It just comes from misunderstanding how the language of mental states works. But you're very strongly attached to this view, and people don't like it when I say that's not how the mind works, that's not what the mind is. But I love being a heretic. So here's the misunderstanding that people have. We'd like to let other people know what our perceptual systems are telling us, what's going on in our brain. Telling them which neurons are firing doesn't help, because all our brains are different. But we can convey information about what our perceptual system is telling us by telling people what the normal causes of that would be. So let me give you an example. Suppose I take some acid and I see little pink elephants floating in front of me. I don't advise this. And I say, I have the subjective experience of little pink elephants floating in front of me. Let's try and explicate what we mean by that. What I really mean is that what my perceptual system's telling me, my perceptual state, is like the perceptual state I would have if little pink elephants were floating in front of me. Now, philosophers have completely misunderstood this. They've invented things called qualia, and philosophers think there's an internal theater, and there's little pink elephants in this internal theater. And if you ask what they're made of, the philosophers say they're made of qualia. They're presumably made of pink qualia, and sort of elephanty qualia, and maybe floating qualia, and maybe the right way up qualia, but that's what they're made of. That's just a dumb mistake. It comes from thinking that I have the subjective experience of something works like I have a photograph of something. If you have a photograph of something, I can ask, where's the photograph and what's it made of? The words subjective experience don't work like that at all. The words subjective experience are to indicate to you, I'm trying to tell you about a perceptual state I have that I know to be wrong. And the way I'm going to tell you about it is by describing a state of the world such that if the world were in that state, this would be correct. That's the best I can do to tell you what's going on in my perceptual system. So subjective experiences are hypothetical real objects. That's why words like pink and elephant apply to them, just as they do to real objects, that would explain my perceptual state. And what's weird about them is they're hypothetical, not that they're made of qualia. So can a computer have a subjective experience? Well, I'm going to give you an example of a computer having a subjective experience. So I get a multimodal chatbot. I'm still under time. I get a multimodal chatbot, and I'm trained up so it can talk and it can see things and it can point. It's got an arm. And after it's trained up, I put an object in front of it, say point at the object, it points at the object. No problem. When it's not looking, I put a prism in front of its lens. And then I put an object in front of it and say point at the object, and it points over there. And I say to the chatbot, no, that's not where the object is. The object's straight in front of you, but I put a prism in front of your lens. And suppose the chatbot then says, oh, I see, the prism bent the light rays, so I had the subjective experience that the object was over there, even though it's actually straight in front of me. I claim that if the chatbot says that, it is using the word subjective experience correctly, and it's using them in exactly the same way we use them. So multimodal chatbots, when their perceptual systems go wrong, already have subjective experiences. When their perceptual systems go right, they have experiences, objective experiences, and when they go wrong, they have subjective experiences. So there is no sentience defense. It's not like there's something chatbots couldn't have that we have. Multimodal chatbots have sentience. They already have it. There is no kind of protective stuff that we've got that they could never have. They're just like us, except that they can share knowledge much better. And so we should be very scared. And that's it. Okay, I'm done.
Well, my goal is, after the last talk, is to give you a distillation of a talk I thought I was going to give, assuming I learned anything from the last talk. So, anyway, my institute was formed in 1992 when I left NIH. And we're approaching our 34th year next year, which is also part of a major expansion we're making to include adding clinical research to all the basic science that we've been doing. Now, Steve asked us to talk about the future, and I thought it was important to put timelines into perspective. As you can see from the slide, it's only 140 years from Darwin's publication of The Origin of Species to our very first sequence of the human genome. In fact, the key event came in 1944 when Avery discovered that DNA was actually the genetic material. So I was born in 1946, so fundamentally everything that's happened in this entire field, changing from thinking that proteins were the genetic material to genetics, to actually having our genetic code and trying to sort out how it codes for each of us individually, has all happened during my lifetime. So it's kind of hard to predict what could happen 100 years from now when you see the world totally changed in reality and conceptually in less than a century. Science moves very slowly. It moves in punctate changes. Most scientists don't move things forward. Most science funding doesn't move things forward. As in all fields, a tiny percentage of people make leaps that change the thinking that existed before. And I argue frequently, it's not like people like myself and my colleagues are making brilliant breakthroughs. We're just proving that the ideas of the past were misguided and wrong. Proteins were not the genetic material, even though Avery could not get the Nobel Prize because biochemists on the Nobel Committee for even past Watson and Crick still believed that proteins were the genetic code, and this could not be correct. So changes come slowly, they come cumulatively, but based on punctate events. Now, I've had a unique view of this question, what is life? And I'm only the second person to give the Schrödinger lecture on what is life. He gave the first one, but I have a unique view. I've been using the lens of genomics, which didn't exist in Schrödinger's time, even though he predicted that the genetic code could be as simple as the Morse code, because everything at the time was sent through the telegraph with dots and dashes. I was talking to the late Francis Crick about this, and he goes, that was such a trivial idea. Everybody knew it at the time, apparently except biochemists. So I had an idea based on trying to put mathematics together to deal with the thousands of human genes we were discovering by randomly sequencing RNA clones. And I thought this might be useful for actually assembling the entire genetic code. I'm not a mathematician. I've worked with great mathematicians that made me look really smart by hiring them and doing this. But this was a simple mathematical problem of taking true randomness of random pieces of DNA that we got 500 letters of sequence from, and finding the overlaps until the complete genome could be put together. This ended up being the first one in history because nobody was thinking of this approach. NIH and other government agencies were funding multi-decade-long approaches to try and sequence E. coli or yeast or other things. Ham Smith, the Nobel laureate, and I wrote a grant to NIH proposing this method in 1994, and it was turned down with extreme prejudice. I even wrote a letter to Francis Collins when we were 90% done and said, look, it's really going to work. NIH should fund it just so it won't be embarrassed when we announce it. He sent me back a letter saying, we're positive it won't work. Take your best shot. And it became part of world history a few months later, followed by a large number of other genomes. Agencies and companies were forcing massive amounts of money on my small institute to sequence genomes, because at the time we were the only ones to do it. The Human Genome, quote, project was a 15-year, initially $3 billion project. It ended up being more than 15 years and more like 5 billion when you see what the British government and the Wellcome Trust put in. We thought the idea we had of using the mathematical shotgun method could change that. We sequenced the human genome in 9 months for a mere $100 million. Steve Scherer was one of our key colleagues on publishing this very first human genomes and subsequent ones. And it really shocked the world. But we've used the same randomness to characterize entire environments. So this book that came out late last year was how we sailed 65,000 miles around the oceans, just taking samples of seawater, filtering out the organisms, and shotgun sequencing them. And this simple idea from taking samples from a sailboat, we discovered more new species than the rest of history of science put together. Just asking a very simple question. And the impetus for this was a paper that was published by eminent scientists in the Proceedings of the National Academy of Sciences that the ocean was almost devoid of life and diversity. And as a sailor and a swimmer, this didn't make any sense at all. And just the very first sample we took off of Bermuda, we discovered thousands of new organisms and several million new genes. But with all these genomes we were sequencing, including the human genome, most of the genes were of unknown function. We couldn't find anything in the databases related to them. AI has been helpful with protein structural predictions, but it's not very accurate. You can find things in structural categories, but it's still hard to sort them out. And so early on, we decided to take the Feynman's approach of his actual quote was, which is different than the internet, and Caltech sent me a picture of the blackboard where he wrote it down. What I cannot create, I do not understand. We actually built a code to write this in the genetic code in genomes. And so we simply reversed this process. DNA sequencing is taking biology and digitizing it. So we decided to go the other way, start with the digital code in the computer and rewrite the 4-base genetic code to try and create a new life form. We started with something very simple. We started with the Phi-X174. It was the virus that kills E. coli that Fred Sanger sequenced that led to his second Nobel Prize for developing the method, but it was the first DNA virus sequenced. We tried just making the pieces of DNA and putting them together. It didn't work, so we had to develop error correction methods because the methods for synthesis are inaccurate. And this was the actual first experiment. These clear spots are where we put DNA in. It was taken up into E. coli using the cellular machinery, made the viral particles. The viral particles kill the E. coli, spread to other ones. And so the clear packs are where the bacteria was killed. So we call this situation where the software builds its own hardware. We just put in a piece of code. It made a biological entity, a lytic virus that killed E. coli. Well, this piece of DNA was very small. What we wanted to make was something that was over 1 million letters of genetic code. The tools for doing this didn't exist, so we spent several years developing the various tools to allow us to write the genetic code and assemble it into these large chromosomes. So that was half the battle. The second half of the battle was booting up the genetic code. And to do this, we used our knowledge of the cell. We had a recipient cell, but it had a genome in it. But we knew from sequencing the genome what restriction enzymes it had in its genome. Restriction enzymes are the enzymes that kill and chew up foreign DNA as it comes into the cell. So we made sure that we did not, in our design genome, have any genome sequences that would be recognized by those enzymes. In contrast, we put in designed enzymes into our genetic code that would recognize the DNA in the cell, chew it up, and eliminate it. So it's like taking one of your bodies and possessing it by putting in my genetic code into it. That's what we did. The genetic code was read and produced an entirely new species by hijacking the cellular machinery. So this led in 2010 to us announcing the first synthetic life. I'm having the same problem that Avery and others had. There's huge groups of biochemists that don't think you can have synthetic life until you put a bunch of proteins together in a bag and they create life. This is how all code works. We just write the code, put it in the code, it creates everything biologically for that cell. This caused quite a ruckus. We did a press conference in Washington, D.C., and President Obama announced a new bioethics commission to review the ethics of creating new life forms. The Pope weighed in the same day and said Dr. Venter did not create life. That's only been done once. He modified one of life's engines. It's not clear which engine we modified. It must have been the Ferrari engine. So for the first one, we modified the genome of an existing cell because if we couldn't do that control experiment, we couldn't do this next experiment. So we wanted to design a whole new species from scratch. And we assumed that what we were reading and taught that biology was essentially all known. So we had a contest at the Institute. Several of us designed what we thought would be the gene content to create a new life form. We made several of these versions. Not one of them worked. And so this took over a decade because we had to randomly add back genes until we could get a living cell. It was trial and error, and AI wouldn't have helped because this was all unknown. Out of a genome with 473 genes, 179 of them that were essential for life were of unknown function. So roughly a third of biology of actually essential for life in the most minimal cell was completely unknown to science. So it's been an interesting challenge over the last several years. And fortunately, people around the world got very excited about this. Also, the new protein predictions from AI have helped. And we've solved roughly half of these, working out what the function is. And there's a consortium of hundreds of scientists. We're down to 78 unknowns, working around the world on our minimal cell, trying to work out these unknown genes do. So this is the smallest genome. Extrapolate it to your own bodies, your own genomes, into standard of science and medicine. We know far less than even a single function for half of our genes. And most genes have multiple functions. So we are at a very primitive stage of knowledge of our own genome and our own genetics. This group is publishing more and more papers on solving these, but it's actually a new tool that people are starting to realize that we can recapitulate, in theory, all of evolution. So the minimal cell codes for a very funny cell. It's a large, blubby, balloon-like substance that sort of divides by blubbing off pieces like a balloon would. So a number of groups were working to add genes back responsible for normal cell division that every one of the hundreds of trillions of cells on the planet do constantly. There were 2 known cell division genes. They did not solve it. It took those plus 3 genes of unknown function to restore normal cell division to the cell. People have added motility genes, so now the cell will walk around the plate. So we can add more and more to this basic life form to understand steps in evolution. In theory, we should be able to convert this into a eukaryotic cell. We should be able to convert it into a multicellular species and really understand the steps. But the biggest limitation are all the genes of unknown functions that are essential for life. So I covered the process of this in Life at the Speed of Light. It was called that because we now were digitizing biology, and the notion was if we sequence something on Mars, instead of sending it back in a multi-billion-dollar rocket ship, it could just be sent back as a digital wave in a much shorter period of time. And we could recapitulate it just by making the genetic code and then booting it up on this planet. And we've tried experiments with NASA, taking samples in the desert and showing we could send these to NASA headquarters and back and make the organism and boot it up. So it's very feasible. We also use this. We got with the FDA the first synthetic flu vaccine produced, and showed that we could just send it through the internet. And the notion for this, when the H1N1 flu broke out in China, the Chinese government would not share the clone outside of China. There was also a separate outbreak in Mexico City. The mayor called me and asked me to help, but he couldn't send us samples. The US wouldn't allow them in. But some Chinese scientists sequenced the flu virus and posted it. We just downloaded the code, remade the flu virus, and so we were the only source for the CDC and vaccine companies for that virus. We turned it into digitally making a new vaccine, which we sent to Novartis as an email, recapitulated it there, and FDA approved this not only as a new approach, but as the first synthetic vaccine. This led to the RNA vaccines that we all used during COVID, in fact. I also argued it be used for digital and distributed manufacturing because somebody could have a device plugged into their computer. You just download the code and the device would just print out the vaccine instead of having to wait for it. So we're still waiting for that to catch on. Because my genome was the first one sequenced, a lot of people analyzed it and told me of all the childhood diseases I should have died from and things, and it's still going on. We had a great meeting with Steve's team today. One of the predictions was that I had changes in a gene called ApoE4 that, according to the scientific literature, greatly increases the risk for Alzheimer's disease. So being concerned with this and believing the literature, which was a mistake, I asked my friend head of neurology if they would do a brain MRI and a amyloid PET scan. And he said, we can't because you don't have any symptoms. Our medical system is all based on solving symptomology, not based on family history or anything else. I finally convinced them to do it. They're very concerned that it would adversely affect the rest of my career because they believe the genetic data and that they would find amyloid and changes in my brain. They were very shocked. They called me a few days later and said, you have a brain of a 40-year-old. There's no amyloid. There's no indication whatsoever of Alzheimer's disease. My wife said, no, you're wrong. He has a brain of an 18-year-old. But we've now, this became the start of human longevity because it was clear what the genetics community was producing was a very incomplete answer. And that, well, NIH was funding sequencing millions of genomes, the British government did, lots of places did. Very few people were looking at phenotypes along with the genome. And so I set up a clinic for normal people to get intensely phenotyped so we could start to understand how our code worked. This shows a normal brain MRI. The hippocampus is this yellow area. That's where you see the most deterioration, as well as the ventricles with somebody with Alzheimer's disease. And it's very predictive. We've now, to make a long story short, have screened hundreds of people like me that are heterozygote for the ApoE mutations. Not one of them had any brain changes whatsoever. Homozygotes for ApoE, it was thought to be an absolute deterministic outcome for Alzheimer's disease. We see a large number of people. I have 3 good friends in their mid-90s that are homozygous for ApoE4, that their brains look like the 73-year-old. You can see a very large normal hippocampus. They have no indication whatsoever for Alzheimer's disease. We do see a rough with homozygotes, roughly 80% of people that are homozygous do have some brain changes associated with Alzheimer's, but not in a direct, absolute causal fashion. So it's like the BRCA1 and BRCA2 stories. When those discoveries were made, many physicians would tell women that had a BRCA1 positive test that they had a high 90% chance of breast or ovarian cancer. In fact, it's a 50-50, a lifetime chance, unless you have a family history. So we're trying to change this by getting detailed phenotype data that can be linked back. And one of the biggest changes, again, was a mathematical approach of taking the MRI data and further processing it. So my friend Anders Dale developed this at UCSD. It's called restriction spectrum imaging. So it turns out tumors have slightly larger nuclei than normal cells. They're larger, they have more water in them, but the water is less mobile. So it gives off a totally different MRI image. It may be less mobile because it's interacting with DNA. It's not clear, but it gives a very bright signal. You can see the orange area here. So here's actually a study on prostate cancer doing biopsies versus the restriction spectrum imaging, where just from a straight MRI using this mathematical tool, the tumor showed up very dramatically. Now, this affected me personally. I had elevated PSA levels. I had 2 rounds of biopsies and normal MRIs and was told that I was cancer-free. As I was setting up my clinic, I decided to test all the equipment, and I was testing the MRI, and my radiologist told me that I had 2 very high-grade prostate cancers. This was less than a month after being told by the best of medicine that I did not have cancer. I had surgery right away, and I've been cancer-free for 6 years now. And had I not just randomly gone and gotten screened when I did, we probably wouldn't be having this conversation now. So looking only at people that are asymptomatic, we find high-grade cancer in roughly 5% of everybody 50 and over, tumors that they don't know they have. But they're most all stage 0 and stage 1. And they're 100% treatable and curable in all but the rarest of cases. I'm just giving you a summary of things because limited time. I'll talk more tomorrow about some of this. MRI is basically the only way to determine what your organ fat levels are. And this is very important because if you have non-alcoholic fatty liver disease, you'll probably need a liver transplant to stay alive. We find elevated liver fat in 23% of everybody that comes through across age groups. So normal is 4%. We find some huge numbers. Here's a couple showing 35% fat. It's almost like they're human foie gras. Sorry for that analogy, but so 23% against all age groups. I was going on a 30-mile bike ride with world's expert on organ fat, and we stopped for a rest 15 miles through. He goes, by the way, doing this exercise won't change your organ fat. Only calorie restriction will. I said, well, why in the hell you didn't tell me 15 miles sooner? So calorie restriction, alcohol restriction is the only way to actually reverse organ fat, but it's something that nobody could ever see before, detect before, until it caused outright disease like severe liver disease where you needed a transplant. These things are all detectable early and totally correctable because it's before symptoms appear. This MRI is fantastic. This is non-contrast MRI, just using the mathematics. The entire vasculature lights up. in a spectacular way. One of the surprise findings is we found all these blebs, brain aneurysms, and we find them in now 1% of the population. So it means at least a couple of you in this audience have a brain aneurysm. If you're over 60, you probably don't have one. The population is skewed to those under 50, because if you're over 60, you've already died from your brain aneurysm. Almost everybody knows somebody that's died from a brain aneurysm. I'm about to turn 78, so I'm free from all disease statistics. So it's but they're totally treatable in a 15-minute outpatient procedure, just through catheterization. So there's no reason for 1% of the population to potentially die from these. These are just a few highlights of what we find. We find 53% of people who think they're healthy have a major disease that they're completely unaware of. In most cases, we find it early enough to do something about it. But the goal would be to, the grand challenge I'll give to Canada, if you adopt these approaches, my calculations for the US, it would save the US economy $2 trillion a year in medical expenses by detecting things early. But the problem is the entire medical infrastructure is based on reimbursements for solving symptoms and traits. So if you have a cancer and it's causing pain, you're probably at least already at stage 3 cancer. Cancers don't just all of a sudden appear where one day you have it and the next day, the day before you didn't. These can be developing over decades. They're easily detectable with this new method, with the exception ovarian cancer and soft tissue cancers are currently very difficult to detect, and I'll talk about that in a minute. We're trying to couple this with genome findings, so we do whole genomes on every individual. We're about to switch that, collaborating with Steve to do complete phase diploid genomes. But even so, the data is increasingly useful for making some degree of predictions and associations with health. It's very early on because we know so little of the genome. We couldn't work out why I had high-grade prostate cancer. So retrospective detailed analysis of my genome showed that I have a mutation in the testosterone receptor. So testosterone receptor is one of these triplet repeat genes. So it's a series of 3-letter codes that determine the expression of the testosterone receptor. If you have 18 or more of these repeats, you're sterile. You can get all the testosterone in the world, it won't do anything for you. So the fewer number of repeats, the more you express the testosterone receptor. I have the least number reported in the literature, so I greatly overexpress the testosterone receptor, and something that wouldn't have surprised anybody when they knew me at a younger age. I was bald by 21, but having an endocrinology study, I actually had a very low testosterone levels. Well, they didn't measure the receptor. You don't need much testosterone if you have massive receptor expression. The same works with estrogen and every hormone, but we only measure one hand clapping in medicine. So I was put on exogenous testosterone because I had low clinical levels. So that was like pouring gasoline on the fire. If I had a slow-growing prostate tumor, that's why they would have accelerated growth. We cultured my prostate cancers. They grow 10 times faster in the presence of testosterone. So it was very clear why they were developing. Now, the good news or bad news is people who live to 50 and beyond half of you will die in the next 25 years, between 50 and 75. And the two leading causes are very clear, and they've been clearer for a long time. It's cardiovascular disease and cancer. The third leading cause are accidents, including now all the accidental overdoses, people falling downstairs. So people want to have control of their lives. This type of data is one way to get control because it tells you all the things you didn't know about your body that you can't see, but with these new tools of science, we can see and tell you you can do something. People try and diet and exercise. Exercise is probably the single greatest thing anybody can do if you want to live past 70. The third leading cause of death are accidents. Most of those are falls of people falling. Car accidents are way down, which is why organ donation is way down. This is in the US, the deaths last year, and all the ones in red are things we detect with our non-invasive screening. But one of the things I really like to point out with this, human longevity to me is not based on trying to live longer. You can see there's this huge discrepancy between what I call the health span and lifespan. While women do live longer generally than men, they spend the last decade or decade and a half of their lives in ill health. And that's what can change very simply with these early discoveries. My philosophy is very simple. If we can keep you from dying from heart disease or cancer, and you exercise so you're less likely to fall, you're going to live a whole lot longer than you would have otherwise. This is sort of people born in different eras. So 1900s, the average died by 57. In the category I'm in, I was born in 46, so I'm not quite in that group, but I'm beating the averages already because I'm about to turn 78. People born today will, in theory, live longer, but the big question is, by doing preventative health, and as we get more knowledgeable about the genome, we get more knowledgeable of how to do pre-symptomatic testing. We know we can shift these curves to the right. We don't know how far. I think the key thing about shifting them to the right is changing the health span, not necessarily just the lifespan. The last thing we're doing is in the clinic at Human Longevity, there's a huge difference between the number of males and females that go there. Women will send their husbands, boyfriends, sons, but are reluctant to go themselves. The answer we hear more often than not is, I prefer not to know. And that's easy to understand with the symptomatic medicine that's practiced today. But if there's ever a case where knowledge is power, this is it. If you detect something early, you have the knowledge and the power to do something about it and having it not be a death sentence. To help deal with this, part of the Institute versus Human Longevity, which is a for-profit company, we're starting a new Women's Center for Transformative Health, which is part research and part applying all these techniques. The biggest single expense in all of this is the 4 specialist radiologists that it takes to read the whole body MRI and the brain MRI. I have no doubt that that can be totally accomplished with AI. There was a wonderful study that was just finished at Caltech where they took slides from tumors where they knew the outcome. So the slides were 10 years old. Pathologists could not look at these slides and predict who would develop metastases and who wouldn't. They trained an AI system to look at it. The AI system is 85% accurate in predicting, just based on the same data, which ones will go on to develop metastases and which ones won't. As you heard earlier, it's very hard. You can't go pick apart what the AI is seeing to do those decisions. So we don't understand. People can't see anything looking at these slides that the AI systems are seeing, but they're consistently really accurate. Pathologists, as I said, are best 50-50 with this. Women's imaging is so much more complicated than men. Ovary is the most difficult organ to diagnose tumors in because of ovarian cysts, because of follicles. My colleague who actually led the team, Sam Levy, to sequence my full diploid genome, develop a circulating DNA test that uses DNA methylation to show the tissue of origin, that detects early ovarian cancer, also detects early pancreatic cancer. And the test field they use is recently diagnosed type 2 diabetics have a much higher risk for that. So we think applying AI will make a big difference. The last thing I want to close with is in 2007, and you can see Steve was a key collaborator on this, was the first human diploid sequence. So you actually get a set of chromosomes from each of your parents, in case you were confused by that. But when the quote genome was sequenced, it just shuffled all that data together. So we were able to separate my parental chromosomes by actually sequencing individual sperm cells. It would work with egg cells as well, but egg and sperm are each haploid, so we could get that information. My two chromosomes differed by 0.5% from each other. And it's called a compound heterozygote. You could have variations in the genes that you got from your mother, but a totally different set of ones you got from your father. And this becomes very important for tracking heritability of ovarian cancer, breast cancer, et cetera. We looked at over 1 billion data points from all the genomes that we had sequenced. And without knowing anything about the sequence, you can see these points that go all the way down. Those are places where the genome cannot tolerate any mutations. So those are probably mutations that would cause spontaneous abortions. We can look at other genes. We can look at all the transmembrane domains. And if you get mutations in the transmembrane area, for example, changing from a neutral amino acid to a charged one, it'll spit that protein out of the membrane and be loss of function. So simply looking at the code, and this was even before AI tools, we can predict just looking at the code whether something's likely to be a disease area or not. The second to last message is mice are not mini humans. Human cells in culture are not mini humans. You can see in this Venn diagram, there's very little overlap with genes essential for life in each of these different conditions. So genomics is at a very primitive stage because people were just looking at single nucleotide changes in now millions of genomes. All of a sudden, the world is shocked to find out that it only explains about 40% of heritability. So, for example, your height, varied study shows that 70 to 9% predictable genetically. All the data from millions of genomes, they can't find the variants to explain more than 40% of the genetics. So we showed in the studies with Steve sequencing the diploid genome that 22% of all the variation exist in structural changes that in fact include 75% of the genome that's in variation. So for the last 25 years, geneticists have been looking under the lamp for their lost keys. They find them 40% of the time. And so by knowing what we don't know, we can start to look to a different future. Thank you very much.
Thank you so much, Craig. Wonderful, all-encompassing talk. Kathryn North now, and we're going to continue probably till 7:15 or so. If, and Kathryn, you have full time.
Good evening, everyone. And thanks to Steve, your team, and what an amazing event here, but what amazing few days that you have planned for us. I'm going to talk much more around single gene disorders that are associated with some of the most severe diseases, particularly affecting children. And as I think we've heard from our amazing two previous speakers, that there have been so many collective efforts, not only in genomics, but in information, that are really changing the way that we're conducting and looking forward in terms of human health. It's been quite reassuring to me to see that each of the three previous speakers has actually looked a bit at history rather than just looking at the future. Because to me, the past, I want to provide to you a bit of perspective about what's been happening in human health related to genomic medicine. But it also, I think the perspective and the context is very important. I trained initially in pediatrics and as a neurologist. And what I found we would diagnose patterns in kids, but we couldn't really determine the underlying cause. We could make a diagnosis that was usually descriptive in about 10% of cases. And there were very few therapies that were particularly useful for so many of those kids. When I trained in neurology, epilepsy wasn't thought to be a genetic disorder, and there are people in this room that have proven that that's not the case. So it's, I think, just going back a little bit to where I started. This is where I did, during my medical degree, I did a research year, and literally my mum typed my thesis, and we cut and pasted it to put it together. In 1993, this is what I did my doctorate on, this wonderful computer. And by that time in my neurology training, I'd figured that genomics and genetics, well, there was no genomics at that stage. Genetics was probably where this field was going to go in terms of really understanding the basis of so many severe neurological disorders. So I went to Boston, and because here Lou Kunkel had just identified a couple of years earlier the first of the neuromuscular genes, the gene for the most severe form, one of the severe forms of muscular dystrophy, Duchenne muscular dystrophy. And I worked with Alan Beggs, who's also here in the audience, to do genetics training. Now, I got a great experience during that time because we were sent to, or allowed to go up to Maine, to the Jackson laboratories, where there was a 2-week course on human genetics. And this was during the time when the human genome was being sequenced by, and as Craig has talked about, a massive international effort going across many countries. And I never forget, we did afternoon clinics. Sometimes we saw mice, sometimes we saw humans, but we did a computer clinic where they talked about this newfangled thing, the World Wide Web, and that it would likely be something that would make a difference in medicine and in human genetics. And then there's the Human Genome Project, and what an amazing effort that was, and how it has changed, how we approach, particularly in my field, how we think about not only the treatment, but the prevention and early intervention of disorders. When I came back to Sydney and I set up a neurogenetics service, I had one of my particular interests was in neuromuscular diseases. And in these early years of clinical practice, as I said, we could come up with less than 10% of diagnoses. There are a handful of genetic causes known, and each one of these kids has something that is different, but they all present with severe weakness or difficulty walking. If you're a parent and you bring your child that can't walk into the clinic, you want to know what has caused this. Why can't my kid walk? What should I be expecting for the future? What are the risks to my other kids or the other members of my family, and what can you do about it? And there was a time when we were really helpless and couldn't answer many of those questions. I'll give you an example. This is a little girl who I first met in the year 2000. When she was born, she was very weak and had to be supported for both feeding and breathing in the neonatal intensive care unit, and she was in ICU for 9 months. Now, at that stage, our diagnosis was based on muscle biopsy, and she had what, I'm not going to go into the disease, called Nemaline myopathy, where she had these inclusions in her muscle. And at that time, there was only one known gene for Nemaline myopathy and a handful of genes for neuromuscular disease in general. And, you know, we got her out of ICU. We did a big international study that showed that if you could get these kids off the ventilator, they tended to eventually be able to walk. But then over the next decade, we searched for the cause. And this was a time when we were doing extensive muscle studies to look if we could identify what the mechanism is. This is what normal muscle looks like. It's a thing of beauty. It's very regular, so it can contract. But in this particular disorder, you can see it's very disorganized, and there's these collections within the muscle so that it doesn't work appropriately. In my lab, we were sequencing the genes one by one as either we discovered them or they got discovered, and it was called a candidate disease gene approach because at this stage, we were only just starting to get access to the background reference human genome. We did this for 10 years, and the parents were very keen to have another kid. We found no abnormalities. This gene Nebulin is really, really big. And at that time, it would have cost us $7,000 US, which is about 13,000 Australian dollars, to send it over to America just to get that one gene sequenced. So we did it by an indirect method and still had no diagnosis. The parents decided to take a chance and to go on to a second pregnancy. So this family ended up with 2 very handicapped kids. It was a great burden on the family. The mum had to stop working. The marriage broke up. This is not an uncommon thing when you're dealing with kids with quite severe disability. Thank God the future looks better in terms of what we can do with this group of disorders. So it was very expensive, as Craig has pointed out. It's probably, I always say $3 billion, but it was probably $5 billion to sequence that first human genome. And there's really been 2 elements that have really made the change over this last 20 years. And the first is that the cost of sequencing has come down dramatically. So I'm going to take you forward now to around 2013, where it was still pretty hefty at around 10,000. This is in US dollars. But it was starting to get to the point where we could think about using it, certainly from a research perspective, if you're in a very rich institute, or how we would think about it in clinical practice. The other thing was technology is changing. Now, back in the 90s, I had one of those brick mobile phones, and now you think of the phones that you all have on your desk or in your bag that actually carry half your life around them and are about thousands of times more powerful, or even more than that computer that I did my doctorate thesis on. So this is where it made a difference, where we could actually start to include genomic sequencing into our workup. And I'll acknowledge that this work, I had 2 PhD students, Daniel and Monkel, who'd gone to work at the Broad Institute, which is a very rich institute, but also was really a hotbed in terms of looking at genomics in disease. And very happily, they wanted to collaborate with our group because we'd assembled hundreds of kids with neuromuscular disease. And not surprisingly, this was family number 1 on my wish list of can we solve this case. So this is the family tree, the 2 unaffected parents and the 2 affected kids. And it was family number 1, and I must say we identified a gene actually really quickly because we'd done a lot of work on understanding what sort of gene it would be, and we found variants in a not previously described disease gene called lyomodin, where I said I would eat my hat if it wasn't the disease gene, because lyomodin is actually one of the genes that organises these thin filaments in skeletal muscle. So the pathology made sense. But that isn't enough. We confirmed that both parents were carriers of 1 copy, and both kids had 2 copies. But to make the case, you have to find additional families. You have to share your data with others, and you have to put the story together. So what would we do in these days? Well, I emailed my mates. Now, that sounds like it was a little bit time consuming, but we have this amazing international collaborative network around neuromuscular disease where we were all working together to try and solve these diseases, which is the only way when you're looking at these genes one by one as we were. So I emailed my mates, and over the next year, around the world, we'd screened over 540 additional cases of this rare disease, Nemaline myopathy, and we'd identified 17 patients from 12 additional families. What we found is that the vast majority of these kids died of this disorder. In actual fact, it was only that they were the girls who lived. And it's not because we are fabulous clinicians in Australia, but it was because they still expressed some of the protein, which was very different to the others, and thus they had a milder form of the disease. So this is just an example of a very long odyssey. It took 15 years to get that answer from the time that it was born. And in collaboration with the Broad, we studied this cohort over the next 4 years and look at the difference that being able to do that sequencing made to all those families that didn't have answers. We ended up solving around 90% of the cases with Nemaline myopathy, but we're overall hitting around a 40 to 50% success rate. And let me say that there's over 500 genes identified for that variety of neuromuscular disease, so that acceleration in gene discovery is really incredible. So I'm now going to talk about the future, but in the context of the present, because what we regard as extraordinary today is going to be commonplace tomorrow. So I just want to give you a feeling for how I think this is starting to pan out by telling you what I'm really excited about at the moment. I see that genomics, it's a disruptive healthcare technology to be able to roll something of this complexity out at scale, not just the sequencing of the 3 billion base pairs, which is equivalent to 1,000 copies of War and Peace, which if you've piled it one on top of the other is equivalent to an 18-story building. So it's a lot of information in every genome. But it's also the compute, and it's been amazing hearing from Geoff and Craig in that thinking of how the technology is evolving in terms of how we apply it to human health. Now, I've been talking about rare disease diagnosis down the bottom here, but where I see genomics is going to figure in our health system is around our life course, because we really want to transform what we do into, as Craig was saying, prediction and prevention and early intervention. because, that is what is going to give us healthier trajectories, but also healthier lives. And I think that quality of life is incredibly important. This is happening in the healthcare system, and since that time, the price of sequencing has come right down. Where, currently it's maybe, depending on what your vendor is and the deal you do with Illumina, it's probably sitting around, somewhere between $100 and $1,000 per genome. But it is predicted, and I actually think this might be an underestimate, that more than 60 million patients will have their genome sequenced in healthcare by next year. But we're also looking, and I'm going to touch on this right-hand side of the circle, because we talk about rare disease diagnosis and how do we then look at prevention when it's looking at these very severe disorders that affect 1 in 12 births, are the major cause of death in children under 1 year of age. And at any time in my hospital in Melbourne, in Toronto SickKids, it's estimated that around 1 in 4 kids in hospital at any one time has a rare genetic disorder. So there's many of them, and collectively they are not that rare. And what has been great is Craig has actually given you a vision into the personalised predictive genomics, but also how it should be altering our healthcare. So let's start at thinking about prediction and prevention and early intervention, but how we do it at speed. First genome was 11 years. And really, when we're looking at clinical practice, say 5 years ago, it was taking, months to actually get a result back from your genomic test. So let me introduce you to who's a patient of Kathryn Howell, who's in the audience, and had a normal pregnancy, was born after a normal pregnancy, but started having quite severe seizures on day 3 of life, which were very difficult to control. And here he was transferred into the neonatal intensive care unit. He needed assistance with his breathing and with his feeding. And as Mrs. Mum said, it was a terrifying time, multiple seizures, with the baby turning blue. Now, happily, he lives in a time where it is possible now to return a genomic result in the matter of days. And he was a part of a study where he was able to have ultra-rapid genomic testing, so that in that first couple of weeks of his life, which is critical because the more you have FITs, the more brain damage that you do, it uncovered a very rare condition which actually has a targeted treatment so that the seizures respond. And now, so is relatively seizure-free. He has some developmental challenges, but is doing pretty well. And I believe when he came into clinic a couple of months ago, he was showing his doctors his kung fu moves. So this, to me, this is one of the miracles in, certainly my work lifetime, that you can, from what wasn't considered to be a genetic disorder, it's now through the work of, again, people in this audience like Ingrid Scherer and Helen Cross. We now know that, I think we discussed yesterday, over 1,000 different causes of genetic causes of epilepsy. And Steve has been hosting us for the last couple of days with this great partnership, because I talked about partnerships to helping get a diagnosis in the olden days, but we've now brought together IPCHIP, or an International Precision Child Health Partnership, that is bringing together the expertise across Toronto, Boston, London and Melbourne, where we're working together to how we, one, look at this prospectively. How do we make these early diagnoses? How do we then make sure these kids are getting targeted therapy? Epilepsy was the first and a great example of what we can do working together, because within a relatively short period of time, around 12 to 18 months, we were able to enroll 100 babies, found a diagnosis in around just under half of those, but it impacted their treatment in around half of the patients. So that's that's what we want to keep increasing those numbers and making that difference. In Australia, our big focus is how do we provide an equity of access to this sort of work, and how do we take something like this nationally. So we've just completed a study of over 450 infants that involved every neonatal and paediatric intensive care around the country, with a whole different range of disorders, from liver failure or immune deficiency, and we were able to provide a diagnosis while those kids were in ICU in over half of the time, and often these kids wouldn't get a specific diagnosis. The test was highly cost effective, but it was hugely impactful that we could do a time to report in 3 days for a national approach, and we were really impacting the management of these kids in over 75% of cases. So this is one case where we have a nationalised health system, and certainly in our state, the day that the research funding ended, they said, the neonatal intensive care physician said, this is like crack cocaine, we cannot do without it. And so it was actually funded immediately within our healthcare system. So prevention is better than cure, and this is so true for everything that we do in healthcare. But it's a real bugger to try and model it economically to convince governments that this is where they should be spending their money because of the health systems that we have. So I really want to look at how we shift healthcare from treating disease after it arises, which is costly and often ineffective, to actually preventing them based on what's going to be a combination of both genetic and environmental and lifestyle factors. So going back to my little kids with neuromuscular disease, and I'm going to focus on this disorder, and this disorder is called spinal muscular atrophy, which is like an inherited form of motor neuron disease that affects between 1 in 6 to 10,000 infants. It's one of the major causes of death in infancy, and it involves the spinal motor neurons that come off the spinal cord and plug into the muscle to give it its juice, so to speak, in terms of stimulating the muscles, don't develop properly. So these little kids have normal brains, actually very bright and intelligent, but in its most severe form, they usually die within the first year or two of life from respiratory failure and swallowing difficulties. And I'm going to introduce to you because she has a major program of work named after her in Australia. And she was diagnosed at 4 months of age with spinal muscular atrophy, and she died at 7 months of age. And her parents, they thought they'd done everything that they should do in having a healthy pregnancy and a healthy child. But they didn't know about carrier screening because it wasn't something that their doctor had told them about. And once they found that they could have prevented this from happening, they lobbied the federal government, they met with the health minister, and he was appalled and actually launched a large national research project called Mission. Now, this is about to come out, it's either out or about to come out in the New England Journal, where we actually conducted this study around the country. We did it during COVID, we did it remotely, and we had been told we needed to demonstrate that we could get equity of access to this around the country. So everything was dynamic consent. People did cheek swabs and sent things in. But instead of testing for just 3 disorders, which is current standard of care in Australia, we actually looked at all the disease genes for a number of different disorders where a couple might want the choice to alter their approach to a pregnancy if they had a high risk of one of these disorders, so causes of physical and intellectual disability. And things that are the most common ones that have been screened for currently are cystic fibrosis, Fragile X mental retardation, and spinal muscular atrophy. But what we found doing a over 9,000 couples were that, in actual fact, 1 in 50 couples are at higher risk of having a child with one of these severe disorders. And these common ones, cystic fibrosis, Fragile X, and SMA, only account for 20% of that. So we're not actually screening for all the things that we could to prevent. So, but it's a big thing. It's we're working with government to see how it might be possible to expand this so that it continues beyond a research study. But when we're talking in our country, which is small, 300,000 births a year, we really need to be thinking, and it's the barriers are economic, of how we're driving this sort of preventative approach into practice. So newborn screening, I thought it was very funny actually that this was in 2012. Do you want to know my future? It's all in your genome. But there's certainly, it's possible. It's certainly possible to do genomic testing in the newborn period, but this is still very much in the research space, because the important thing here is if you detect something in the newborn period, it's most acceptable if you can do something about it, rather than, as has been quoted by, a lot of preventative testing just creates the wealthy worried well. And we really want to make sure with newborn screening, and currently as it is that we can make a difference. So this is very much in the research phase, but there's big international consortia. This is just showing where studies are going on, and we bring all those groups together so that we can learn from each other, because is society ready for this is the other big question. Then really finally what it comes down to then is how we use all of this information to actually better targeted therapies for our patients. And again, I thought this picture paints a thousand words. So this little boy is the oldest of 3 children, and he has that severe form of spinal muscular atrophy that I talked about. He was born before there was any effective therapies. He's quadriplegic. He's never walked. He's ventilated. Has to be tube fed. His younger brother was diagnosed during the pregnancy with spinal muscular atrophy. But at that time, and this was in 2016, the treatment was first made available. He actually started to receive 2 monthly infusions into his cerebrospinal fluid of what's known as an antisense oligonucleotide therapy that actually provides or upregulates something that compensates for the gene that is missing. Now, still affected, but much more mildly than his brother, and he can actually walk by himself. Their youngest sister was also diagnosed in utero with spinal muscular atrophy, and she received a single dose of gene therapy at 11 days of age, and she's basically unaffected by the disease. This is I guess this is to me the miracle of my career, for a gene that was first identified in 1995, and to see the difference that this makes. But I'll give you my second last example is that, similarly for dwarfism, which can be life-threatening because you can get compression of the spinal cord, but there are these new life-changing treatments now after international clinical trials. And this little boy here has actually been on a drug that actually alters the underlying cause to the most severe, most common form of dwarfism, the one that Tyrion Lannister had in Game of Thrones. And he now has a normal growth rate, which prevents those medical complications with spinal compression. But now as we do trials and start to give it earlier, you can actually completely normalise the growth in this particular form of dwarfism. But why did I put this up? because it's then about access, and it's costly. These treatments are incredibly expensive. So again, it's going to be a societal decision of how we apply this amazing technology and use it within the healthcare system. My personal view is prevention is very important in how we think at a societal level. And then finally, there's the really cool stuff. Stem cell medicine is really starting to become a potential reality. And since Yamanaka in Japan was awarded the Nobel Prize in 2012 for the ability, we could take blood cells or skin cells from any of you in the audience, we could reprogram so that they were like embryonic cells, like an original human egg that can differentiate into any tissue in the body. And this is the basis then for the potential for stem cell medicine. And you can differentiate into three-dimensional organoids across a whole different range of tissues. And it's great for modelling and understanding human disease in human tissue, but also to start looking at gene correction and how you might start to treat disease or use it for regenerative therapies. And we're particularly looking at it for heart disease, for kids with holes in the heart, or for children that have had severe congenital heart disease, which affects 1 in 100 kids, that can, despite multiple surgeries, can end up with heart failure. And this to me is just a great example of having scientists working side by side with clinicians to really look at how you might apply this into practice. This is a heart patch made from James's human tissue and grown. And this is not James's heart, this is a lamb heart, where we're starting to look at how these tissues integrate into the beating heart, to look at how you could wrap that around that heart to increase its force of contraction. That brings big challenges from the data, and I'm going to finish with the data challenges, is that we're not just looking at the genome, and as Craig has put out, we need to be looking at what you're exposed to, your exercise. It is as complex as Google Maps in pinpointing what a cause is to take all of this stuff into account. And then dealing with that amount of information. And we go from, all the discoveries the human genome has available to, I love this quote, there were 5 exabytes of information created between the dawn of civilization and 2003. But that much information is now being created every 2 days. So how do we interpret that? Obviously, the importance of global data sharing, I think, is incredible. We talked about the importance of international collaboration in terms of how we work together, but data needs to be open source and shared, so we can take that data from millions of people to apply to improve that prediction for one individual. And I think Craig gave some great examples where you really need to know the frequency of certain variants associated with different forms of cancer. So I think these 2 gentlemen who've spoken before me sort of demonstrate the breadth and the creativity of thought that needs to go into this. And I love this quote from Jeff. To deal with a 14-dimensional space, visualize a 3D space and say 14 to yourself very loudly. Everyone does it. I got him to explain that to me, so you should corner him afterwards. But it's you need to think outside the box. And Craig, who has just really spent his life looking so broadly and thinking outside the box. But I think it is this characteristic. I don't know if optimists or pessimists are right, but the optimists are going to get something done. And that's the principle of the Global Alliance for Genomics and Health. And Heidi Rem's in the audience, who's the chair. I've been vice chair and involved for many, many years. But this is looking at how do we safely and responsibly share clinical and genomic data for the betterment of human health. It's now involving over 100 countries around the world. It has led to tools that are used where we're starting to connect these databases, but around the world so that we can get to rare diagnoses much more quickly. And that's been incredibly successful. But I think the answer is going to again be, how do we reach and link all of this information so that we can use that to bring it to predict to the individual? And that's where I shall leave it. But thank you so much for your attention.
Wow. I'd like to invite our panelists, our speakers and panelists to the front, please. And ask everyone to stay a little bit longer. Please just have a seat at the front. Janice and Darren. So this was just absolutely incredible, all three presentations. I invite Darren or Janice, would you like to ask a question? And then I have one question.
Go ahead.
Then I would like to, no, you go ahead. Then I would like to invite our speakers if they may have a question for each other. But please, one of our panelists go first.
Maybe I'll start us off then. Darren, is that all right? First of all, thank you for these three absolutely extraordinary presentations. We're all so lucky, and thank you to Stephen for organizing this. Across all three these, there were, I think, two common themes. One was data. As we go forward, how important it is to you, James, how important it is to Jeff, what you're going to do, and Kathy, as you talk about building this network. And right behind data was compute. As you think forward over the next 25 years, when we face these extraordinary energy challenges in the context of accelerating climate change, which connects back to preventive medicine, how do each of you think about solving that problem? Because that is ubiquitous. The answer, yeah.
You have to use solar energy.
So when you look at those projections, the pace at which the demands for compute will grow will far outpace what the most optimistic, and you have to be optimistic, but what the most optimistic predictions for solar will be.
Well, you also have to use nuclear energy.
Yeah.
And what people like Sam Altman are now considering building nuclear power stations for data centers.
I actually heard the other day that Bitcoin it takes up about 20% of the world's energy supplies in cryptocurrency, and that there are thoughts about charging, if we think of the cost, and particularly to look at sustainable energy sources, then we also need to look at who should pay for it. And so starting to take that look as well. And then I'm hoping Craig's going to come up with some super form of energy conserving data management, and what I'm also interested in what quantum computing will do from that perspective.
I guess that means I have to answer it. Well, one of the things we've tried to do in energy is modify cells or create cells to be far more efficient photosynthetically to actually produce lipid molecules to replace oil. And it works on a scale right now, but it's not economic enough to replace oil and gas. But when you think about the ability to write the genetic code, there's a fundamental change that's happening that I didn't get a chance to talk about, but it's a company called Avery, it's working in San Diego, is using computer chips actually to write the genetic code. Each pixel on the computer chip they can synthesize DNA on. One of the reasons the first synthetic cell took 10 years and cost tens of millions of dollars was we had to chemically make these molecules by hand. And we had to do one experiment at a time. So you had genes, you have to then test that. With this new synthesis, if we had that, it would have been a few months to do the 10 years of experiments. because we could have made 10,000 iterations and then just screen for which one's going to produce life or which one's going to produce energy more efficiently. So biology is much the source of solar, and solar biology is even better because it builds its own renewable system versus having to use precious resources to create solar energy. So I think we live in a biological world. We can turn to biology for solutions.
Thank you.
So I was really taken by the precision medicine approaches and the new therapies, which I just astounded by. But it also raises a question in that it's super impressive to get these therapeutic effects like the spinal muscular atrophy. But one issue is how do we go from showing a handful of people on a slide to treating more broadly? I mean, some of these are some of the most expensive medicines to develop. I mean, it also applies to sort of cellular therapies and genome editing as well. How do we go from just a few people on a slide to actually treating people in our own neighborhoods, because I think that's a big bottleneck. And it will take not just sort of medical advances, but venture capital perhaps and funding and just the clever ways of thinking about things. So I wonder for your perspectives on both from a sort of medical perspective and a big picture society perspective, how can we get these great treatments to more people?
So just a small question. I think that now is a very important time for us as a society to be thinking ahead of what this could look like. You're right, the treatments, spinal muscular atrophy to me is a very good example of what we can do. But as a society with the cost, these treatments, the gene therapies cost us in millions. That's why I also bring in the preventative strategy, which is very cheap. We need to be really thinking how we do this as a society. And we're just at the very beginning for these very precision therapies to be available. But a lot of them, they're accessible in clinical trials, but then the bills are in the millions or the hundreds of thousands per patient. And I don't think we can cope with that financially as a society. And that's why we really need to be thinking backwards and thinking right back to the, to where these things start and how we're promoting prevention.
Thank you. Are there any other ideas from a sort of bigger picture perspective of how maybe this can come about?
Well, there's no reason that drugs need to cost what they do. Cystic fibrosis may be one of the best examples. We have one of the discoverers here, the mutations that were discovered here in Toronto. There's now very, very expensive drugs finally for treatment. They're fantastic treatments. But it's the same thing with the new GLP-1 agonists that could eliminate obesity as, if not a disease, as a major cause of disease in this country. But the cost structure is completely misguided, as is most of the money that goes into paying for medicine. But the argument is that companies need to make absurd profits or they won't discover new drugs. But there's balances on it, and drugs are about a quarter the cost in Canada as they are in the US. So you have less to complain about up here, but it's still, they're not things that people can generally explode. If you want to really look at the distant future, and it's probably more than a century away, most genetic diseases can be edited out of the germline of the genome. And we can have societies that are largely disease-free, but we know about 1% of the information we know to do that intelligently. The argument is, for example, manic depression. If that's treated as a disease and you edit that out of the genome, you'll wipe out human creativity. So most human genes don't just have a single function. Some of these rare genetic disorders that we heard about so nicely, maybe the rare exceptions. It's very easy to eliminate Tay-Sachs disease or ataxia telangiectasia from the population. But it turns out it's a very slippery slope. All the surveys that have been done of these higher order purposes, in my view, most couples would use them for fairly trivial traits like height, like hair color, eye color, the muscle genes that determine fast twitch or slow twitch, so it could determine what kind of athlete they're going to be, versus being focused on eliminating diseases that people die from in childhood. So we're not a mature enough society to do this. So the first germline engineering probably won't take place for a very long time. If local politics in the US show anything, we're going backwards, so we may never get there. But those could be the long-term solutions.
So I have a question. Goes back to my opening slide on the future of Homo sapiens. And I've been waiting a generation to ask these two gentlemen this question, but we kind of talked about it on the way over. So each of us is a result of a sperm and an egg coming together. And within that cell, you have the genomic information. And the cell itself is smart enough to divide and lead to the development of an individual like any of us in the audience that looks almost identical to our counterpart parent, with some mixing, of course, but you can often look at photographs, and many of us in this room study the biology behind the mechanisms of development, these developmental processes that when they go awry, it leads to disease. So my question, maybe to start with, to Craig, is if we had enough genome sequences, could you take the genome sequence and predict what the phenotype would be fully? And we talked about an example on the way over. Please share that. And
if we have enough genome sequences together with the phenotype data to combine the two, because the genome on its own is virtually impossible to do that. Working with Franz Ock, who developed Google Translate, worked with us at HLI to use AI to see if we could predict what somebody would look like from their genetic code, using a number of traits of things that were known to affect nose shape, et cetera, hair color, eye color, skin tone. And we were very effective with this, but people are incredibly critical of our study on this because they didn't like the answer. And it gets down to what you think your genome would code for if we were using your genome to predict what you look like. And it turns out it's sort of a cross between pre-adolescence and post-puberty is the age that the genome seems to predict versus how people look like when they're 50 or 80. But there's algorithms that can easily age the photos. But we thought this would be a rapidly adopted new forensic tool, for example, with the close to 1 million missing children in the US a year and things like that, for tracking down what they would look like. But there was just so much resistance to it and thought that it was trivial. AI's improved about 3 orders of magnitude since we did that study. So it should be a fairly simple thing to do as an example, but that's a physical trait versus predicting whether somebody has a photographic memory or aphantasia like I have with no photographs at all. These things could affect the education system. They could change all the tracks. So it's not just about medicine, it's about who we are, how societies function, how we get educated, how we educate future. So.
But you, on the walk over, you said that the prediction from your genome in the PNAS paper was actually quite accurate to when you were 16 years old or so, right?
Yeah. Everybody that we showed the my genome prediction, the pictures too, So that looked exactly like published photos of me when I was an early teenager.
So when Jeff heard that, he was kind of surprised, I think. My question to Jeff is, how far is AI going to take us in deconvoluting the understanding of development and evolution, perhaps?
There's also questions about whether we want to do.
Okay.
So psychology's got a
He's good.
history of, I forget the name of the guy, but he averaged the face of lots of criminals to get what a criminal looked like.
Mm-hmm.
And that probably wasn't a good thing to use. So
It was very racist in its application. Yeah. That's why.
So we have to be very careful about how we use these technologies,
Yeah.
particularly in authoritarian societies.
So we heard then.
But I can't resist. So how do you think about this 25 years from now, Jeff? Because that's all about institutions and politics and society. How do you think about who makes those decisions, which are good and which are not?
Well, actually, I know I got my own mic. I got on. I'm not sure we'll make it to 25 years from now. We seem to be going, well, we'll see what happens on November the 5th. But there seems to be a big movement towards authoritarian societies. China, for example, just refused to sign something saying they wouldn't put AI into the control loop of nuclear weapons. I guess he hasn't seen Terminator. So there's lots of other threats. So with sort of gene editing technologies and with AI and precision medicine, we could have a wonderful future. But we've somehow got to deal with these terrible risks. And the worst risk of all comes from the combination of AI and gene editing, which is that you can relatively cheaply now make something like COVID. Some years ago, there was a Japanese cult that released sarin on the Japanese subway, on the Tokyo subway. And what they would do now is use AI tools, particularly the one recently released by OpenAI, to design nasty viruses. And I think that's a threat that may not allow us to last 25 years.
So that's a threat that's been with us for the last 30 years.
But it's getting much cheaper.
The cost is immaterial. It costs a few $100s to make. So my lab made 30 different versions of COVID for the pharmaceutical companies to test their vaccines against. It took 1 week and a few $1,000s. Any virus that's been sequenced
no, but this is new, designing new viruses. So with AI.
I think AI could probably design a new computer virus, but I don't think it could define a new human virus because we don't even know the functions and most of the genes are unknown. So I think that's a hypothetical fear that's very far off. We have much more real ones. Flying into Canada, what is the first thing you're met with is a face recognition system that now is being used more and more. We can easily move in the world, as I've shown, between the digital world and the biological world. So we can improve by probably 6 orders of magnitude face predictions by using all this data from the face recognition algorithms that by presumably all of you, if you ever left the country, are in the computer systems. Any of us that came in, we're now in the computer systems and along with other phenotype and physiological data. So this world's upon us, and it's a question of how it's used. China's using genomics to track minority populations and discriminate against them. In the global sharing, China's a one-way door. They take genetic data in. It's not allowed to leave the country. So international things only work if there's 100% participation, in my view. So I agree with a lot of problems. I have no doubt about the next 25 years. They may be very ugly. I'm more concerned about the next 4 years. They could be really ugly. But,
Okay. We're gonna stick a few more minutes. I'm gonna ask a rapid fire question to each of the 5 people at the front. You know, Karen, what do you think is going to be the highest impact paper in Nature when we look back 25 years down? What's coming up the pipe?
Not to be put on the spot or anything. I think genetic therapies for disease, big area, and so genome editing, but I think used in the right way. So I think there, especially at the moment, there's very little justification for germline genome editing because almost always you can select embryos to not need to do any editing and still have something perfectly healthy. What would be the highest impact ones would be for diseases that have no other option. Something like muscular dystrophy is a good example where there's a lot of good progress, but that's also an issue where the body has a lot of muscle and it's not about correcting the genes, it's about getting enough editors into the body. So it's a multi-part problem of getting everything across the right tissue. So I think some of the biggest parts, the things that I will handle would be these sort of genetic therapies and done in right ways that solve delivery problems and efficacy problems and resulting clinical benefits. That would be my perspective.
Sorry for putting you on the spot.
For other people,
please do not hold it against our next submission to Nature.
I mean, just randomly other things. I mean, engineering microbes to degrade plastics and things. I mean, there are lots of global problems that can be solved through science, we hope.
Thank you. And Kathryn, an experiment we should or shouldn't do?
This discussion has been quite depressing because I think what it throws into stark relief is that societal values and the ethics of what we do should probably be first and foremost in our mind. I'm obviously excited as a geneticist and a pediatrician by being able to solve some things that were completely insoluble, but we need to be, it's what's happening at a societal level that is, I think, where we should be focusing more of our laser-like attention and also speaking out about that. And I know that's not quite, because, I think we can do stuff, but it's what we're going to do with it.
Right.
So when my guys come and say, I want to do a newborn sequencing study, I just go, but where is that going to go? How would we apply that?
So you're positive about the future, very positive.
I was. and until about 7 o'clock.
Janice, over to you to turn it around.
Well, I'm very positive about the future, although I very much understand why Jeff is worried. I don't dismiss that. But I think there are 2 really important points that I would take away from all of this. Second of all, we need to do a much better job than we have over the last 50 years in joining up science and social science and law. Because if it's bolted on it at the end, then Jeff confronts the problem that he rightly talked about, that Chinese scientists won't sign on to one of the most important worries that we would all have, which is, really, really smart AI-driven autonomous nuclear weapons. That's a world that none of us want to be in, but we haven't done a good enough job about joining that up. Mm-hmm. And then I think the second comment I would make is the best antidote to pessimism is the kind of international collaboration that you talked about. But again, not just, we need the genetics, the geneticists in the room with the social scientists. So, if you think back, and I was just talking about this to a group of scientists, to the role that the International Panel on Climate Change played, there was no consensus whatsoever when they started their work, but they were broadly multidisciplinary. They brought together whole groups of people, built those networks, and published research, which really changed public understanding. I don't think it's not government's job, it's all of ours
And Jeff, any closing comments? And I'd like to hear just quickly your, I know of your career and the importance of fundamental research very early on, and how that.
Sorry, I didn't hear.
I know of your, so I want to hear your closing comment, but also if you can just also comment on the importance of fundamental research very early on in your career trajectory.
Yeah, so if you look at the pharmaceutical industry, a lot of that came out of people like Watson and Crick discovering the structure of DNA. The research done with X-ray crystallography in the 1950s and 60s, that wasn't done in order to create a pharmaceutical industry. Maybe it was, but basically it was funded as pure research. If you look at the neural networks that are now the basis of AI, for many years they were just a curiosity, and almost nobody thought they were going to overtake classical AI. They were funded because people were interested in how the brain might work. They didn't discover how the brain worked, but they did create a technology. So it's basically curiosity-driven basic research that leads to really big breakthroughs.
Thank you. That makes my job easier. And Craig, closing words?
So I think the big test in all of this is going to be see which populations, which governments step up to change the healthcare of their country by doing pre-symptomatic testing, adopting these new tools, adopting early predictions. There's no reason for cardiac deaths and cancer deaths to be the leading cause in this country and every other country around the planet. That could change virtually in a year if countries decided to put their resources, instead of paying for post-symptom care, to move it steps earlier. They're the ones, in fact, governments and third-party payers. CT scanning came out in the 1970s, and clinics opened up around countries, storefront clinics for people to get whole body CT scans. CT scan is just X-ray, and so it could not distinguish between a cyst and a tumor. So there had to be ultrasound, biopsies. Health companies, the governments didn't want to pay for all these extra tests, even though they were life-saving in many situations. They applied a term from incidentalomas. Saved lives, but we found these other incidental findings. Modern mathematical tools, the new MRI changes it. So the incidental rate, the misdiagnosis, is less than 0.1% right now, with close to 10,000 people going through. So it's a different world today. That there's no reason to hide behind it, except everybody in the medical establishment gets paid by solving riddles and symptoms instead of treating health of a nation, of a population, of individuals.
Okay, with that, thank you very much. I am very, wait. I am very, very positive based on the attendance and the participation. And I teach a first-year graduate course at the University of Toronto in medical biophysics, and we have an ethics class course that all of the students have to take. Our students are our future, and they think in a much different way than probably most of us in the audience. So I see that as a very bright future. And I have a slide, I didn't include it today, but essentially what I wanted to show you today is that as a species, we can essentially do any experiment we want now. And the experiments we do are only limited by our curiosity, right? And so the questions we pose are based on our environment and the influences of how we've been brought up and our beliefs. And the main message that I would like to get across today is that it's very important to continue to support basic, pure, fundamental research and the people that are doing it, and train them to think in a very, very broad way. And you heard a lot about that today. So that's the message I would like you to take home. And I want to thank our speakers and our panelists so much for participating. Thank you very much.